Hey everybody! So today you are going to learn about how DNA gets transcribed and processed to make polypeptides and proteins. So a couple things to remember is the normal way of proteins being made is you go from DNA, which makes it gets a copy made of it of RNA, and then that gets translated into a protein. So genetic information is stored in the DNA molecules or RNA molecules, and here's just a visual of here's your DNA transcribed into RNA and then into your polypeptide, which is a protein. So what are some differences between DNA and RNA? So DNA is double-stranded, as you can see here. RNA is single-stranded. DNA is only found in the nucleus, and RNA is found inside of the nucleus and outside. You have thymine instead of uracil, and there's only one type of DNA, and there are 3 different types of RNA. So how do we make mRNA? So first of all, the hydrogen bonds are broken between the 2 strands. The template strand is called the non-coding strand, and then the non-template is called the coding strand. The gene is transcribed on the template strand. So this right here is RNA polymerase. Notice that DNA polymerase makes DNA and RNA polymerase makes RNA. And it also, if you notice, goes from the 5 to the 3. Okay, so 5 to 3 is how it's synthesized, just like DNA polymerase. And so if it's making it in the 5 to 3, that means it's actually reading the gene in the 3 to 5. I know, it's confusing. Okay, so after the primary transcript is made or the immature transcript, a couple things are done to make it mature. So the first thing is that we add this poly A tail, and that's a bunch of adenines stuck on the end of the tail, stuck on the 3 end side of messenger RNA. And what that does is it increases the stability of messenger RNA. So when messenger RNA leaves the nucleus, which is what it also helps it does, it goes into the cytoplasm, and there's lots of enzymes that are dangerous and break molecules down. And so the longer the poly A tail, the more stable it is, and the more it will be transcribed and made into a protein. Okay, the other thing that happens is you have a cap, a GTP cap on the other end, and that also protects the transcript and it helps it bind to the ribosome to translate it. The third thing that happens is that introns, and they're part of the transcript that do not code for the protein, get what's called spliced or cut out. And so you can see the introns are these. And what happens is those get spliced out, and the exons get linked together to form your mature mRNA transcript. I think drawing this picture would be great, just so you have a visual of what these words actually represent. Okay, here's just another picture in a slightly different way. So you have your 3 ends, so you would have your cap. Actually, I guess it flipped here. And then so here's a 5 end with the cap, and then here's your poly A tail on the 3 end. And then as you can see, the introns get snipped out, and you end up with just exons to get to your mature transcript. So something that can happen while these modifications are going on is what's called alternative splicing. And so that's the process of cutting out introns and connecting exons to get a messenger RNA. So that sounds really familiar, but what can happen is different messenger RNA can be made, mature messenger RNA can be made depending on the exons. So for example, here is your messenger RNA, here's your 4 exons. And so in here, 1, 2, and 3 make protein A, but then if you have 1, 2, and 4, so let's say 3 gets cut out because of the type of protein it encodes, then you get protein B. So this is a way for genes to make multiple proteins, is just different exons and introns get cut out. Okay, so students often get confused with the differences and similarities between transcription and DNA replication. So how is transcription like DNA replication? So nucleotides are added in the 5 to 3 direction in both cases. RNA polymerase and DNA polymerase both read in the 3 to 5, and both processes happen in the nucleus. So that's how DNA replication and transcription are similar. Now, transcription is unlike DNA replication because in transcription, you only transcribe a small stretch called a gene. In replication, the entire chromosome, all of the DNA is replicated. RNA polymerase is what is adding the nucleotides and instead of DNA polymerase because they're making RNA. And the product in transcription is a single strand. So mRNA or messenger RNA, the product in DNA replication, remember, is a double-stranded molecule that's identical to the previous one. Okay, so like we talked about before, there's three types of RNA. You have messenger RNA, which is right here, and that is the instructions for building the protein. And so the message gets taken from the DNA to the ribosomes. The messenger RNA is separated into what's called codons, which are in threes. And the codon codes for a specific amino acid. And then you have transfer RNA, which is right here. And transfer RNA has what's called an anticodon. And that anticodon is going to be complementary to the codon. And then down here would be an amino acid. So each transfer RNA has a specific amino acid that is going to go to the ribosome and match up with its complementary codon. And then the last type of RNA is basically what ribosomes are made out of, rRNA. There is a large subunit and a small subunit. And then with DNA, there's only one type. Okay, so we've talked about transcription. We have our messenger RNA. It's left the nucleus, and now we're going to the ribosome to make our protein. So there's three main steps, and we'll talk about each of them in more detail later on in the lecture. So you have initiation, which you initiate something, you start. You have elongation, that's where you build the most of your polypeptide, and then termination when you're done. So let's start with initiation. It would be a great idea if you drew some pictures because I think that helps you learn and helps you kind of visualize this crazy stuff that's happening in your body. So messenger RNA start codon, which is right here, binds to the small part of the ribosome, okay? And so they're grouped, like we said, in triplets called codons. And each codon is going to correspond with a particular amino acid, okay? And many amino acids are encoded by more than one codon. So I'll show you that in a second. But like AUG is always the first codon in every single protein, the thousands and thousands of proteins that are made in your body. So once that codon gets hooked into the small subunit, then the transfer RNA with the complementary anticodon, okay? So if this is AUG, then this would be UAC, okay? And that's gonna match up, and that's the amino acid is called methionine, or meth for short. Okay, and then after that, the last step is that this large part of the ribosome is going to hook on to the small, and we're going to get going. So here's another picture of how the anticodon is going to match up with the codon on mRNA. Okay, next part, elongation. Here's where all of the amino acids get put together. So as the messenger RNA goes through the ribosome, kind of like a cash register receipt kind of thing where it goes through, the transfer RNAs are going to deliver the amino acids in the order, this is important, specified by the messenger RNA. So I think of transfer RNAs as taxis. So they have directions, right? Their anticodon is the directions to get them to the correct place on the codon on the mRNA, and they're just delivering their amino acid. And then if you notice this methionine, right, this is the transfer RNA that delivered it, and once it delivers it, it leaves and goes to get another amino acid. Okay. When you have enough in the ribosome, peptide bonds, which is a type of covalent bond, is formed between the amino acids. Okay, until you get a whole chain of them. So then lysine comes in, it matches up, and then you're going to get a bond between tyrosine and lysine, and then tyrosine, this transfer RNA is going to get shot out and then come back in. It's just going to keep going and going and going until you have like hundreds of amino acids together. The final step of translation is what's called termination. Just like it sounds, this is how it stops. So if you notice, there was a start codon, right? And that did have an amino acid called methionine. However, when the messenger RNA gets to the stop codon, there is no amino acid that matches that. And so this stop codon causes the ribosome, the two parts of the ribosome, to release from each other, to pop off. The messenger RNA leaves, and then you have a polypeptide, which is going to go through secondary, tertiary, and quaternary structures, if you remember, in proteins. So remember this, the order of amino acids is the primary structure. Okay, so you get a stop codon, that's going to cause the ribosome to disconnect, and then you have your polypeptide chain. Now, a really bad mutation could be if one of these letters in your codon was mutated into what the stop codon is, then it could cause the protein to be half made, which then it wouldn't be functional, and that would cause problems. Okay, so you need to be able to read a codon chart, which is what this is. There's 64 triplets that can be made. However, there's only 61 amino acids, so some of them, as you know, are the same amino acid, even though the letters at the end might be different. So how you read this is if you had CAC, okay, so the first base is C, the second base is A, so you go to where C and A, and then the third base is C, and so then you go over C, and so it'll be right here, it'll be histidine. Okay. Now, if you notice CAU is also histidine. So if this last nucleotide was mutated, it wouldn't have any effect because it would be the same amino acid. Now, UAG is the stop codon I was just talking about. So if that comes up, then the ribosomes separate and you're done making your protein. Okay, so now that we've made our polypeptide, what happens to it? So some of the proteins enter the cytoplasm, and they're enzymes or structural proteins, and they do their job. Many go through the ER, where their secondary and tertiary structure is modified. Okay, so central dogma is kind of is like the basis of biology. Okay, central dogma of something is a big theme in the content. So central dogma in biology is that DNA is transcribed into RNA, which then allows you to make a protein. Now, of course, because there's always deviations, there are differences, and one of them is what's called a retrovirus. So retroviruses go from RNA to cDNA, which is a copy DNA, back to RNA, and then to protein. So let me explain an example. So retroviruses insert RNA. They also insert this enzyme called reverse transcriptase. And that enzyme coming from the virus, so that's a viral enzyme, is going to turn RNA into DNA. Okay, and so then once the infected cell has that DNA, it's not going to know the difference between its DNA and the viral DNA, and it's going to make messenger RNA, and then it's going to make proteins. And what happens is there's a lot of mistakes in this process from RNA to cDNA, right? The reverse transcriptase is not very good at making DNA. And so with those mistakes or mutations, you get mutations in the virus. And that is what makes it so hard to make an effective vaccine, is because the virus is constantly mutating. So once the DNA is made, right, then the viral gene is transcribed and then it makes new viruses. And those viruses are made inside of the host cell and then they get secreted out, or the cell actually bursts. And an example of this is HIV. HIV has a reverse, is a retrovirus. And that's it. And we'll watch some videos in class to kind of show you this. Please draw some pictures and hope you learned something. Thanks.